Hello. Today we will be talking about advanced genetics as it relates to cat coats. We'll cover 4 topics: sex linkage, X inactivation, polygenic inheritance, and multiple alleles. First we will talk about sex linkage. So, in cats as in humans, males have an X and a Y chromosome and females have 2 X chromosomes. If one allele located on an X chromosome is dominant, it can mask the recessive allele on the other X chromosome if that organism is female. If it's a male, it cannot, and very often the recessive allele is expressed because the male does not have a second X chromosome. In cats, this relates to the gene for red coats, which is located on the X chromosome. So, because it's located on the X chromosome, a red male would have a dominant O and the Y allele. Red female would have 2 dominant O's, would be a homozygote. Black male would be lowercase oy, and black female would be oo. So tortoiseshell would be the heterozygote, which would be oo. This is typically only females, but it can be males, which would be o, lowercase o, y. A heterozygote with 2 X chromosomes and a Y chromosome. So here's a picture of what a red cat looks like, and there's the tortoiseshell, the heterozygote female. So why does the tortoiseshell look like that patchwork pattern? Because of X inactivation. During the organism's development, each cell inactivates one of its X chromosomes. Let's look at some to create a bar body. Let's look here. So we have the genotype for a heterozygote tortoiseshell female cat, right? So we can inactivate one of the 2 alleles. So we'll inactivate the black one, and that cell will be red. It'll have the red hairs. Or we can inactivate the red one and it'll have black hairs. So this makes the blotch pattern. It makes it sort of patchwork for the coat pattern. So next we'll talk about polygenic inheritance. In cats, polygenic inheritance is relevant in these white spotting genes. White spotting genes do exactly what you think they do. They make white spots. So cats can have varying levels of white spots depending on how many white spotting genes they have. It exists on a sort of sliding scale. So, for example, this first cat has not that much white. It's a calico tortoiseshell with white genes. This next calico is a lot more because it has more white spotting genes than the last one. Next we'll talk about multiple alleles. You probably recognize this example on the left. It's blood types. But we're talking about cats and, cat coats. So we'll just forget about blood types for a little bit. So the gene that codes for tyrosinase, which is an enzyme that helps in the creation of pigment of any color. It could be like dark black or just like red or whatever. It exists with 4 alleles. So the first allele is the dominant one. It just makes healthy, normal tyrosinase. The second one is messed up. It makes albinism. And these make a mutant form of tyrosinase. This form of tyrosinase is special in that it's dependent on temperature, and it's only active at lower temperatures. So it creates a sort of heat map on the cat where you can see where the lower temperatures are based on how dark it is, like in the paws and the face and the ears. So the normal tyrosinase cat would have just a C because it's dominant, and then anything else. It could be like another dominant, it could be a homozygote, or a bat, it could be Burmese gene. An albino would have 2 lowercase c's, 2 recessive alleles. The Burmese would have a C super B and then another C super B. And then Siamese, just like the Burmese, has the mutant tyrosinase, except it's just a slightly different coat, the S superscript. The Tonkinese is a mix of the 2. It has a mink coat, and you guessed it, it's a mix of the 2 alleles, so CB and then CS. So here's a Burmese cat. You can see the dark areas. The Siamese. Actually, I think I got those mixed up. And there's the mink cat, the hybrid between the 2. They can also be tortoiseshell, so you can have pointed tortoiseshell cats.